Good afternoon. Today we're talking about tricking cells into repairing their own mutated DNA and using the gene editing enzyme CRISPR to repair Fragile X chromosomes. If you don't know what Fragile X syndrome is, it's a genetic condition responsible for developmental problems, including learning and cognitive disabilities. It's also a leading cause of autism spectrum disorders. Children with this disorder might also suffer from anxiety and hyperactive, like fidgeting behavior, and males are more likely to be affected by this because they only have one X chromosome, and Fragile X is caused by a damaged gene on the X chromosome. Females can get this disorder too, but typically the symptoms aren't as severe because females do have a second X chromosome. If you remember from biology, your DNA is composed of these nitrogen bases, and they're represented by the letters A, T, C, G. With Fragile X syndrome, the FRM1 gene has more CGG sequences in it than it should, and it's kind of like putting extra letters in the middle of a word, and the word doesn't make any sense. The extra CGGs in the middle of the gene also cause the entire gene to become inaccessible or methylated. So as a result, people with Fragile X can't use the gene, and then they don't make the FRM1 protein, which plays a large role in development, particularly synapse development, those connections between nerve cells. The lack of communication between nerve cells would then lead to cognitive disability and sometimes seizures among those that have it. Now here's where Harvard scientists used the CRISPR enzyme. They didn't directly edit the Fragile X chromosomes. They used the CRISPR enzyme to detangle, or it's called demethylating, the big FRM1 gene with all of its extra sequences that had become knotted up. So when they unknotted it, this triggered the cell's own DNA repair enzymes to recognize those extra CGG sequences and cut them out, which ultimately restored the function of the FRM1 gene so that the correct protein was produced. This is exciting news for those who suffer from Fragile X syndrome. Although this study was done on cells in vitro, it may be possible down the road to package this specialized CRISPR, like put it in a virus or some other vector, and deliver it to the brain cells of those affected. But this is at least a few years away, and there are other questions to be answered. Also, if you didn't see my pinned TikTok about why two identical mice don't look alike, you might want to check it out because it goes into more detail about how genes can be turned on and off.